like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Welcome to the National University of Singapore Yong School of Medicine Healthy Longevity webinar. It is great having you today, and we are talking about a very important organ today, and that is the brain. Associate Professor Sajikumar Sreedharan is with us tonight, and you might have lots of questions, so please use the Q&A function. First, we are starting with a short presentation by a PhD student of our guest, by Mohamed Zaki bin Ibrahim. And he is going to talk about sex-specific time and activity-dependent plasticity and memory in an Alzheimer model in animals.
Thank you for the kind introduction. As mentioned, my name is Zaki, and I'm a graduate student from Sajikumar Lab. Today, I'll be presenting a recent work from our lab on the sex-specific differences in Alzheimer's disease published in the journal Aging Cell. This work was done together with our collaborators from Nanyang Technological University. Alzheimer's disease is a progressive neurodegenerative disorder that mainly inflicts memory loss. One of its pathological hallmarks is the accumulation of amyloid plaques in the brain. Various factors can increase one's propensity to develop the disease. One risk factor is age. Besides that, evidence suggests that sex may play a role. Women are twice more likely to be susceptible to the disease. It was assumed that this trend arises from the fact that women tend to live longer. However, this assumption is called into question by various statistical studies. Thus, this sex discrepancy could involve underlying biological processes. The aim of this study was to establish the role of sex in the predisposition to Alzheimer's disease and the mechanisms that underlie these changes. To approach this, we use this mouse model with genetic manipulations attributed to the progression of Alzheimer's disease. Thus, when older, these mice tend to have greater memory loss than their non-mutant friends of the similar age. Within this mouse model, comparison between male and female mice can be made. We employ field electrophysiology to examine changes in the synaptic response over time. As neurons communicate via synapses, the ability to modify the synaptic response is a proxy for learning and memory. In fact, the breakdown of synaptic function is one of the early signs of Alzheimer's disease. From the mice, we obtain slices from the hippocampus, a brain region vital for learning and memory. These slices are kept viable in an artificial chamber and will allow us to study the synapses in the brain. From here, performance of synapses can be analyzed from slices of the male and female mice, and to produce heightened synaptic response known as long-term potentiation, 3 different stimulation protocols were used. In this graph, you can see the results of the first stimulation protocol. When compared against the mutants, both male and female mice cannot maintain the heightened synaptic response over time. And you can also notice that there's a decay in the female mice side, which is far more faster and distinct than that of their male counterparts. And this sex-specific difference is also replicated in the 2 other stimulation protocols. Taken together, this set of data demonstrates that synapses in the female mouse has a greater level of dysfunction than the synapses of the male counterparts. Behavior test was also conducted on living mice. The behavior tagging experiment was chosen as it assesses the ability of mice in forming associative memories. revealing more subtle behavioral differences. In these memory tasks for the mice, the female group performed more poorly than the male group in the formation and recall of long-term memory, and this further validates the results seen earlier. And to study the molecular changes in the brain, RNA sequencing was used. This technique will inform us which genes exhibited altered expression levels. Strikingly, we noted that the memory-associated genes were more strongly downregulated in females than in males, while the pro-inflammatory genes were more strongly upregulated. And we also identified several sex-regulated differentially expressed genes in the mice, and this can be a target for future studies. Lastly, immunohistochemistry is conducted on the brain slices to visually quantify any molecular changes. This reveals a high level of plaque formation in females when compared to males, indicating a higher level of disease progression. Furthermore, there is higher inflammation as noted by quantifying levels of activated microglia, the resident immune cell in the brain. Taken together, this report observed a faster decay in the synaptic strength and memory retention in females than in the males of this particular mice model. Collectively, this study confirms that the female mice show a stronger inflammatory response and downregulation of gene expression associated with memory. This may contribute to the memory impairment associated with the disease. And more work remains to be done to better understand how sex differences in the brain intersect with risk factors to influence synaptic function, not only in demented brains, but also during normal aging process. And it also emphasizes the importance of including sex as a variable in many research settings, particularly studies exploring aging and how they impact different disease states. And in the long run, the representation of female biology in biomedical research will advance the development of effective drugs towards promoting women's health. And with that, I would like to acknowledge the people in our lab and our collaborators for this work. And this work is mainly supported by this grant. And thank you so much for listening in.
Thank you so much, Zaki. Very nice results, really nicely highlighting that there are sex differences. So well done. And we will now continue talking about the brain. And we have the honor today to have Professor Sreedharan here. So he is faculty member in the Department of Physiology and research director of our Healthy Longevity Translational Research Program at the Yong School of Medicine at the National University of Singapore. And he is very busy. He is a board member of journals, for example, being part of editorial boards of fabulous international journals, such as Neurobiology of Learning and Memory and the Experimental Brain Research and the Oxford Open Neuroscience, etc., etc. But he is also serving as a F1000 faculty member for cognitive neuroscience since a couple of years, since October 2017. And he won a huge amount of awards, such as the Singapore Neuroscience Association Young Investigator Award, the Faculty Research Excellent Award, and the Graduate Mentor Award from our university. And he receives also lots of research funding from different agencies around the world, including Germany, Singapore, and the USA. So we are very, very fortunate to have you here, and you will talk also about the brain. So the floor is yours, Saji.
Thank you, Andrea, for the kind introduction. And first of all, I thank Brian for inviting to this nice seminar series. This is running for a long time. Let me share the screen. Okay, so today I will be talking about some of the recent results from my lab. My lab is studying functional aspect of synapses, specifically learning and memory changes with aging and neurodegeneration for the past 20 years. So recently we are concentrating how functional aspect of synapses, specifically associative memory and associative plasticity, is impaired with aging and neurodegeneration. First, I would like to invite your attention to a simple graph showing different aspects of memory. Imagine we are making a memory. It may be around 80% or 90% strength. And based on this strength, we will say we made a very strong memory. And here the y-axis represent the memory trace strength, and x-axis represent the passage of time. It may be seconds, sometimes some memory lasts only a few seconds. It may be 1 week, 1 month, or maybe it's a lifelong memory. So the initial aspect of the memory is encoding process. We are making a memory. After that, it is undergoing some kind of molecular cellular process. We call it as consolidation. After the consolidation, the memory will be stored. If it is unable to undergo consolidation, the memory will be decay into baseline level. We call it as trace decay. But in case of consolidated memory, we are trying to retrieve after some time. And if we are able to retrieve those memories 100%, we will say we are having a perfect memory. If we are unable to retrieve those memories, we will say we have some problem. It may be dementia or neurodegenerative disease. For example, in case of Alzheimer's disease, we know that they can make memory, but we don't know which part of memory is affected partially or completely. For example, they can make memory means the encoding process will be intact. Sometimes they can consolidate memory. Sometimes they can retrieve memory. But in most of the cases, if they are unable to retrieve those memory, then memory is we will say there is a problem with memory. So in most of the studies, generally we concentrating on encoding process, the molecular changes happening during the formation of memory. We are unable to identify the changes happening during the consolidation and also during the retrieval process. So we are trying to understand different aspects of memory erasal, memory decay in our lab. And we mainly use a model, an electrophysiological and cellular correlates of memory. It's called long-term potentiation and long-term depression. These are two forms of plasticity, activity-induced change in synaptic efficacy that is considered as a model for storing memory. I will explain this graph. Here, the percentage of synaptic strength represented here, percentage. We have if a neuronal population is undergoing learning process, initially there is no activity happening. We call it as baseline. After that, we are providing an activity either in the form of high frequency stimulation or low frequency stimulation. After high frequency stimulation, the synaptic efficacy goes up and maintains for many hours. Then we call this process as long-term potentiation. And in case of low frequency stimulation, the synaptic efficacy goes down, and then we call it as synaptic depression. It is not mental depression, it's the change in the synaptic efficacy. It goes down and maintained for many hours. And this forms of plasticity, LTP or LTD, we can divide it into two phases, mainly protein synthesis independent early form of plasticity called early LTP, which is analogous to short-term memory, and protein synthesis, transcription and translation dependent process that lasts for many hours, which is considered as a cellular correlate for long-term memory. So when I call early LTP, that means which is analogous to short-term memory, and late LTP means it is equal to long-term memory. And the basic mechanisms of LTP and LTD include, consider two cells, cell A and cell B. In case of LTP, we are providing an activity. In artificial situations, we can provide high frequency stimulation, which consists of 100 hertz, 100 pulses. We provide at different time intervals, maybe 3 times. In most of the labs, they provide with the 10 minutes interval. In this case, glutamate is released from the presynaptic part, and sodium ions that causes the glutamate the presynaptic release of glutamate causes a depolarization in the postsynaptic cell, so that sodium ions goes inside the cell, and along the depolarization, along with the glutamate release, which releases the magnesium blockade from NMDA receptor, so that there is a heavy influx of calcium, which activate kinases. Eventually, it can phosphorylate different proteins such as AMPA receptor, or higher activation of kinases can activate various genes, leading to more protein synthesis. So in short, the synaptic efficacy will go up. In case of LTD, instead of high frequency stimulation, we can provide low frequency stimulation, which is a stimulation consists of 1 hertz for 15 minutes, means 1 pulse per second, which also release glutamate, but less amount of glutamate, which also cause depolarization in the postsynaptic part. Sodium ion senders, because the depolarization is happening because of the entry of sodium ions here. Again, magnesium ion will be removed and calcium ions enter into the postsynaptic cell, but less amount compared to the LTP situation, which activate phosphatases, which can remove different phosphate group. For example, removing phosphate group from AMPA receptors can lead to less number of AMPA receptors or functional synapses in the postsynaptic part, leading to less synaptic efficacy, leading to depression. So this is the basic mechanism of LTP and LTD. And trust me, this is one of these two processes are considered as cellular correlate, widely considered cellular model for long-term memory. And in my lab, we study the structural and functional aspect of long-term memory. Structural means the morphological changes and receptor changes happening with the aging and neurodegeneration. Functional changes means long-term synaptic efficacy and time-dependent associative plasticity, associative interaction, which is the basis for associative memory. And the models that we use in our lab are rats or mice, where we have various animal models, knockout mice for Alzheimer's disease, for aging models. And in recent time, we are also using non-human primates for studying the functional connection between a different synaptic population. So in all cases, we remove the brain and isolate the hippocampus, which is chopped to 400 micrometer thick slices, which are located with electrodes. Here you can see 1 recording electrode and 2 stimulating electrode to stimulate 2 independent pathways to the same neuronal population. And this is the microscopic view of non-human primate brain slice. So you can see here, it's very clear, different pathways can be activated by delivering different stimulation protocol and study for many hours. In our lab, we studied LTP for 24 hours, and we have a beautiful setup by which we can keep the slices for more than 3 days. So today I will be talking about the aging aspect of brain, especially synaptic plasticity changes and functional association. So the first understanding, so what we know about aging at present situation, especially in the aspect of synaptic plasticity and associative plasticity. So we all know that the synapses does change in aging. The number of neurons may be decreased, but still plasticity is present. And this change can be numbers or functional state of synapses. But the aspect of functional state is not studied well. So I will be covering this particular aspect in case of aging. For example, aging rats show faster decay of LTP at hippocampal synapses. You can see here the decay rate of synaptic population after the induction of LTP is much faster in older animals. And if we test the animal in a barn maze, we can see that they show less memory. I mean, they have more forgetting. So synaptic loss is there, not cell loss, but in some cases, all synapses are stronger adaptation at single synaptic level. So we don't know the mechanism for that. Maybe it's a kind of metaplasticity by which individual synapses can adapt with the situation, like aging is a kind of stress. And probably the synapses are undergoing a kind of metaplasticity, and still that show a plasticity that can still code memory. And reduced synaptic plasticity, but still present. So we can't say that aged neuronal population doesn't show plasticity and memory. They can also show memory. They can also encode memory. They can consolidate memory, and they can also retrieve memory. And network dynamic change, but additional circuit can be recruited in aging, adaptation at network level. So this aspect is not studied well. In our lab also, we didn't study it. We are trying to understand more about the network dynamic changes. So in my talk, I will be giving more importance to a specific receptor that is widely expressed with aging and neurodegeneration. It is called P75 neurotropin receptor, which is a low affinity receptor, and which is highly expressed in aging situation. The receptor consists of an extracellular domain, which is a ligand binding domain here, and a transmembrane domain, and an intracellular domain, which is called death domain. The various ligand for activating this receptor is nerve growth factor, neurotropin 3, neurotropin 4, BDNF, and pro-BDNF. So once it binds to the extracellular domain, there is a conformational change in the cysteine domain of the transmembrane that leads to the activation of the death domain. So P75 receptor is highly expressed during the neuronal development, which is a necessity, because high expression of this receptor will fine-tune the synaptic network. So controlling the survival mechanisms of neurons during the early neuronal development, this receptor is very important. And in adult nervous system, the expression of this receptor decreases, but again, it is re-expressed during stressful situations like aging or neuronal injury or after epilepsy. We can see the expression of this receptor is high. So at first, we evaluated the expression of this receptor in our model in various age groups. As you can see here, at 6 weeks, 12 weeks, and 8 weeks, there is a gradual increase in the expression of P75 receptor. Here it is nicely depicted. So at 18 months, we also tested at 22 to 24 months, up to 2 year. The animal means 24 months is considered as the best aging model in case of rats or mice. And these receptors are particularly increased with age in neurons and also astrocytes. And at this age, means at 18 months, we could not see much difference in the expression of this receptor in microglia. That was a surprise for us because neuroinflammation is actually increased with aging. But I'm not showing the data here. At the age of 24 months, actually there is high expression of this receptor. Since the expression of P75 receptor is high with aging, we thought of using a model that lacked this receptor. And we wanted to test what happens to memory and associative plasticity and associative memory in this model. So we used a complete knockout mice, and the young adult is about 5 to 7 weeks, which is a sexually matured animal group. And the aged group is 20 to 24 months. And it has been shown earlier that there is no change in LTP in this mice. For example, here it has been shown in 2005 from Roche group, induction of LTP in this animal showing a normal form of LTP. There is no difference in wild type and P75 knockout mice. But the LTD was highly impaired. So the opposing form of plasticity, synaptic depression, was impaired in case of P75 knockout mice. But surprisingly, this animal shows enhanced memory and greater susceptibility towards stress. And they also have increased forebrain cholinergic neuron. Because of this, they also show high spatial learning and memory. And recently, we have shown that after sleep deprivation, the expression of P75 receptors are increased. So sleep deprivation itself is it's a stress for the neuronal population. And so that P75 receptors are re-expressed in these animals, sleep-deprived animals. So our first question was, what happens to normal plasticity in these animals? Whether we are able to induce normal memory, normal plasticity, long-lasting plasticity in these animals. So we isolated the hippocampus. We used a two-pathway model to study long-term potentiation. As I explained, LTP is considered as a model for long-term memory. And this is a model, this is a cartoon showing the location of the electrode. Electrode means S1 and S2. That means we can provide different kinds of stimulation. For example, in S1, you can provide long-term memory like stimulation, artificial stimulation in one set of axons coming from the CA3 region. In the same time, you can record baseline potential from the synaptic input S2. So this is the two-pathway model. This is the model by which you can study activity-dependent plasticity. That means input-specific plasticity process. Input specificity is the specification of memory because memory is also input-specific. So here, after 30 minutes of baseline, we induced late LTP by providing high-frequency stimulation. As you can see here, there is about 50% potentiation remained up to 240 minutes. That is 4 hours. 4 hour LTP in normal situation in human memory is about a 60-year or 70-year-old memory. And in case of P75 knockout mice, we are able to induce LTP. And compared to wild type aged mice, they show, these synaptic populations show 75% potentiation. So in wild type aged animals, we see potentiation. We are able to induce plasticity in these animal models, but the percentage of potentiation was less compared to normal wild type or P75 knockout mice. So the next question was, what happens to long-term depression? So if we induce a weak form of long-term depression, which is opposing form of plasticity, generally we can induce LTD, the synaptic efficacy goes down and then it goes to the baseline level after 2 to 3 hours, within 120 minutes. But in the aged animals, if you induce a short-term LTD, we see a long-term LTD. There is an enhancement of LTD in aged animals. We also tested it with a chemical form of LTD, which is induced by 100 micromolar DHPG, which is a group 1 metabotropic glutamate receptor agonist. As you can see here, in aged animals, we see enhanced long-term depression. So the plasticity, LTP is a little bit impaired. Impaired means it shows reduced plasticity, but LTD is, it shows enhancement of short-term LTD to long-term LTD. So the next question, what happens to the normal population, whether these neurons are healthy? Yes, they are healthy. So we measured the input-output curve. Input-output curve is a measure by which you can compare the normal synaptic response and also the presynaptic firing rate of wild type aged, healthy, means young ones, and P75 knockout mice. We could not find any difference. So coming to the first topic, whether the synapses changes, yes, they change. Whether the neurons are healthy, yes. In normal situation, those neurons, those are not undergoing memory processes. At basal synaptic level, they are healthy because They show normal input-output curve and normal release probability. So the next question was, what happens to the functional aspect of the synapse? That is the main topic of our lab. Functional interaction means within a particular time frame, weakly activated and strongly activated synapses will cooperate or compete each other to form long-term memory, and which is best explained by a hypothesis called synaptic tagging and capture, which provides a conceptual basis for how short-term memory is transforming into long-term memory within a particular time frame. For example, if somebody is asking you what was the name of the wine that you had last week, probably you may not remember it because there was no need to store the name of the wine that you had last week. But you may remember the name of the wine that you had on your wedding day because it is associated with a strong event. Wedding is considered as a long-term memory. So something associated with long-term memory, the synaptic population will automatically associate and form long-term memory. So the concept is explained here when the cartoon shows a neuron that undergoes memory. When there is no stimulation, no activity is going on there. But when we provide a weak stimulation, there is a structural change in those input-specific synapses. And in this cartoon, I'm showing here a memory-like event, a strong event is giving to the synaptic input, which mark the synapses or tag the synaptic population. In addition, it activates the protein synthesis machinery. It may be local protein synthesis or global protein synthesis. And in condition C, we are providing a long-term memory in one synaptic input. And within a particular time frame, we are providing a short-term memory, which sets a tag or which marks the synapses, but it is unable to synthesize the plasticity protein. But it can hijack or it can share the plasticity protein from the first synaptic input because the first synaptic input is provided with a strong event. This is synaptic tagging and capture. And in our in vitro situation, you can study synaptic tagging and capture by providing high frequency stimulation to the first synaptic population marked here in red, late LTP, which is equal to long-term memory, followed by induction of a weak form of plasticity called early LTP or a short-term memory. Over the time period, the early LTP will be transforming to long-term late LTP. In the same time, the synaptic input S3, which is provided with normal baseline stimulation, show no change. That means the memory is input specific, which is considered, the S3 is considered here as a control input. So what happens to synaptic tagging and capture or associative plasticity in case of aging? So here is the synaptic tagging and capture in wild type aged animals. The first synaptic input is provided with long-term memory-like event, followed by weak memory-like event in synaptic input S2. Surprisingly, we could not see any kind of plasticity, means long-term plasticity in both input. Earlier, when we provided strong tetanization, I mean high-frequency stimulation, we observed a form of plasticity that can maintain up to 4 hours. But here, when an early LTP is provided in the S2, in the second synaptic input, there was no plasticity even in the first synaptic input. That means a kind of synaptic competition is happening. Instead of synaptic cooperation that is shown here in P75 knockout mice, it's a kind of cooperation. Here, the tag said due to early LTP could benefit from the plasticity protein or memory protein synthesized from the first synaptic input. Now the early form of plasticity or memory is transformed into late form of LTP. This is also same with the young wild type animals, which is intact in P75 knockout mice. But in case of wild type aged animals, instead of synaptic cooperation, we observed synaptic competition because there was not enough resources, I mean the memory proteins, to share it with the weak synaptic partner here. So in vitro situation, we have seen lack of cellular associativity. What happens with the in vivo situation? I mean, in behavioral situation. So we used a behavioral tagging paradigm in which we expose the animal to a novel environment. It's a box provided with some novel object. We allowed the animal to play with this object for 15 minutes and then put the animal on a platform. When this is the plexiglass platform here, and the animal is coming down, it will get a mild shock. Here, the mild shock is considered as short-term memory because it doesn't induce any kind of a fear memory. It will forget after some day because it's a mild shock. But the novel environment is something equal to long-term memory because it requires dopaminergic activation. Novelty exposure means there is a lot of dopaminergic activity. It is something like your first time visiting Paris Tower, you will have an excitement there. So you will remember it forever. So here we provided, here you can see the experiment. There is a plexiglass platform. We place the animal there. He is exploring the platform. After that, he will come down. Now, a mild shock is provided. The animal will go up. And over the time period, after 15 minutes, 60, after 1 hour, or after 1 day, or after 1 week, in our lab, we studied after 1 week, we measure the memory. So we again place the animal, but we don't provide the shock. So check whether the animal is coming down and how much time it is spending on the platform, which is considered as a latency, which is a measure for long-term memory. So this is the outcome of behavioral tagging experiment in wild-type animals. As you can see here, the red one, the wild-type aged animal forget the shock thing. Again, it is going down and getting the shock. But in case of wild-type and P75 knockout animals, they remember it even up to 7 days. 7 days is considered as a long-term memory in case of rodents. So naturally, you will be thinking, what are the possible mechanisms? What are the mechanisms embedded in these animals? So there are two possibilities. So one thing, the first one is, does aging impairs the generation and distribution of plasticity proteins? There are many memory proteins generated during learning and memory process. And over the time period, we want to maintain those plasticity proteins to code that memory for a longer period of time. And the second question is, does structural plasticity impairment affect synaptic tag setting? Because synaptic associative plasticity involves two components. The first one is the setting of the synaptic tag, that is the initial fingerprint for making memory. And the second process is the production and distribution of memory proteins. So here we looked on different molecules like ROA, ROC, profilin, and cofilin. And these are well-known signaling cascade for actin dynamics, which is important for synaptic tag setting. And the major plasticity proteins like protein kinase A, MAP kinase, PKM-zeta, and BDNF. So over the time period, we have identified different tag molecules like MAP kinases, CAM kinases, CAM kinase 2, actin network, and TrkB are considered as some of the molecules that mediate the setting of the tag. At the same time, the plasticity proteins are protein kinase M-zeta, CAM kinase 2, Homer 1A, and BDNF. So first we looked on the level of BDNF, and we noticed that pro-BDNF level is increased in wild-type aged animals. So pro-BDNF is actually enhancing a synaptic depression, which mediate the transformation of short-term LTD to long-term LTD, while mature BDNF is critical for the maintenance of LTP. So this explains why we got enhanced LTD in aged animals and which was lacking in P75 knockout mice. In the same time, MAP kinases like phosphorylated P38 MAP kinase level was increased, which is also considered as a molecule for maintaining LTD. And PERG level was decreased in case of aging. With the wild type aged animals, the ERK1 and ERK2 was drastically reduced, but there was no change in P75 young and old knockout mice. So next we looked on the structural plasticity components like ROA and ROC2, which are very critical for setting the synaptic tag. As you can see here, the ROA level and ROC2 level is increased in wild type aged animals, but it was intact in case of P75 knockout mice and wild type mice. At the same time, phosphorylated limb kinase 1 and p-cofflin level was decreased. And also ARG level was decreased in case of wild type aging animal. So in short, synaptic plasticity changes from an excited state, I mean in case of enhanced plasticity to a decreased plasticity level was observed in case of wild type animals. So here it's a summary of the event that is happening with aging. The P75 neurotropin level was increased, the expression was increased. Again, the BDNF level, pro-BDNF level was increased, but there was no change in mature BDNF level. Because of pro-BDNF level, the subsequent subcellular pathways was also impaired, like there is an increase in P38 MAP kinase that enhances LTD-like processes. At the same time, there was a decrease in ERK1 and ERK2 level, which reduces the percentage of potentiation in LTP that we observed. Also, the structural plasticity components like ROA, ROC2, limb kinase, cofflin pathway was also highly impaired with aging, but this was preserved in case of P75 knockout mice. So in total with aging, there is a change in synaptic efficacy, change in associative plasticity, and change in associative memory. Coming to the conclusion, so let us answer these conclusions here. So does synapses change in aging? Yes, we observe that. The change can be numbers or functional state of the synapses. Yes, synaptic tagging and capture was impaired. We observed a kind of synaptic competition instead of synaptic cooperation, which is the representation of the functional state of the synapses. Synapses lost, but in some cases, all synapses are stronger adaptation at single synaptic level. I'm not showing those data, but we found more metaplasticity in aging animals, which is a set of data that we are going to publish soon. And reduced synaptic plasticity, yes, we observed induction of long-term potentiation in aged animals, but the percentage of potentiation was less. And network dynamics change. Over the time period, the synaptic network changes from a synaptic cooperation level to synaptic competition level. And I would like to thank my colleagues. The main work is done by Yong. He is the first author of the paper, which is published in Aging Cell. And this work was done in collaboration with Carlos Ibanez. He is very famous with the neurotropin receptor studies. And I would like to thank the collaborators and also the funding agencies. Thank you very much.
Thank you so much, Saji. What a wonderful talk, really giving us an insight of how our brain works. And I think we try to get that knowledge since a couple of decades or 100 of years, that we think, okay, what we do and what we remember really has a biochemical action, and there is a pathway behind it. But tell me, if I see something, who decides if that goes to the long-term memory or to my short-term memory, that I am remembering that? Also, or I know that, for example, my husband remembers everything and I forget it all the time. So tell me a little bit, how is this selection being made, short and long term? And secondly, why do we see so many individual differences?
Okay, so memory can be classified based on the kind of induction. For example, as I showed in my slide, based on the strength of the stimulation, we can divide memory into short-term memory and long-term memory. If the information, so when we acquire a knowledge, means an information, it undergoes different kinds of processing. So the first part of the memory that we make is declarative form of memory. So declarative form of memory means it need more space, more time to code it as a long-term memory. But sometimes we make memory within a fraction of a second. For example, if we witness an accident, we make a memory, but this memory is completely different than the normal memory that we make every day. For example, if we make some memory means within a fraction of a second, for example, witnessing an accident, it can also activate some unwanted pathways leading to false memory or post-traumatic stress disorder. Like in post-traumatic stress disorder, actually memory is a problem. Memory is haunting us. So that is PTSD. But in case of declarative forms of memory, the best way, in my opinion, the best way is to take time and code that memory, allow the memory component to consolidate and store it as a long-term memory. So the first part is the declarative part of memory, and most of the declarative memories are actually the strength is less. And with the repetition only, we are making it is more with masked or spaced way of repetition. We can make those memories as a long-term memory. So it solely depends on the type of stimulation and also association. Sometimes when we associate the memory component with some other component. For example, when we see something, we can also associate it with the smell. Mm-hmm. So now instead of one component, like visual pathway, when we see something, we are visualizing, our visual cortex is activated. But when we smell something, its olfactory cortex is also activated. So now instead of one neural network, there is two neural networks work together to form your memory. Then the strength is more strong.
And why the inter-individual differences? Maybe also based on several clues you have, you smell something, you see some light, some learn better from watching videos, some reading a book, etc.
It actually depends on the individual difference. I think I can say there is no clear answer for that, why some people are very unique in making memory. So there are many incidents. If you read neuroscience book, you can see so many patients. They are called as patients. They can remember. Sometimes they read only one time, they can remember. It's a kind of photographic memory they have. Yeah. So there is a very famous patient S. It is pronounced as patient S. S for Saji. Yes. So this patient S can remember everything. He remember all the sound. When we see something, he code the memory as sound and some kind of touching experience, somatosensory experience. So it depends on the neural pathways. We can say it's a kind of metaplastic neural pathways they have. So more studies are concentrating in that aspect to study why some people have better memory than others. It will be very interesting to see the difference.
Yeah. Then looking at the different forms of dementia, we have so many. We have Alzheimer, we have Parkinson's, etc., vascular dementia. If you're talking about your fabulous designs of synaptic plasticity, do they all contribute the same? Have you studied yet also other mice models depicting other symptoms or reflecting different dementias?
That is difficult to say because dementia can be because of many reasons, right? Sometimes it may be due to the neuromodulatory differences. In some cases, cholinergic inputs are impaired. Some cases, for example, in case of Parkinson's disease, probably the dopaminergic. Dopamine is also important for memory. It's not only for movement, for memory also, dopamine plays an important role in making long-term memory. So that is affected. So so in our cases, it's difficult to say which one is clearly impaired. So we generally look on NMDA receptor and dopaminergic synaptic input, which is impaired in aging. But in case of vascular dementia, we have a project. We also noticed that dopaminergic signaling is impaired in these animals.
And switching from a disease state, so dementia, switching to something sometimes we have in our hands, and that's sleep. So it was fascinating that we were saying that sleep deprivation is actually changing the P75 expression. So can you talk a little bit more? Is that also the sleep deprivation? What is that, first of all? But also having a little bit less sleep, do you also see then possibly different pathways or P75 expression changing? And is that, of course, reversible? I think I would like to know that. And I think maybe also others here in the audience.
Yeah, actually, I have not presented the data showing the PDE4 level, which is an enzyme that regulates the level in neurons. Cyclic AMP protein kinase A level is very critical for forming long-term plasticity and also memory.
Mm-hmm.
So during sleep deprivation, actually we are activating the same mechanism like that of aging. Believe me, I didn't show that data. So when we have sleep deprivation, in our model, we gave 5-hour sleep deprivation. So we handled the animal, we didn't allow the animal to sleep. And then we tested different memory paradigms. And we analyzed the hippocampal neurons, and we found that cyclic AMP PK level is drastically reduced, which is also same in case of aging. I didn't show that pathway, but it is impaired in normal aging. So one thing is that normal sleep deprivation, when we sleep deprive, we have working memory errors. That's why while driving after sleep deprivation, when we drive, we make a lot of accidents because we could not associate many things at right time and make the right decision. So we can say it can also impair the declarative form of memory because LTP was impaired.
Mm-hmm.
In addition, synaptic tagging and capture, which is the associative memory, plasticity is impaired, and also associative memory was impaired. So we found somewhat similar results like that of aging process. But one good thing with the sleep deprivation, in our observation, most of the changes are actually reversible. Because we gave only if you have a 5-hour sleep deprivation, after 1 week, when you test it, actually it is normal.
Okay, so that's a good thing. And also there are lots of metabolic changes.
That's why once I applied for a grant, and then the comment was, what is the better solution for sleep deprivation is to sleep well.
Yes, absolutely. I fully agree. One short question from me, and then I'm handing over to Danica because I already see there are lots of questions of the audience. Short answer, what is the relation between memory loss and depression? Because it very often co-occurs in humans and in patients. So do we already know what's behind that co-occurrence?
No, actually, during the depression, the neural pathways involved in the depression process are completely different than the normal memory loss. That is the short answer for that, but there is a very complex pathway difference.
Yeah.
The short answer is that the neural wiring is different in case of depression and also in dementia.
Okay, good. Thank you. Over to Danica. What does the audience want to learn?
Thank you for the introduction, Andrea, and thank you for the very insightful presentation, Prof Saji. Here's the first question from the Q&A panel. Are there any neuroscientific basis to preempt significant memory loss?
I don't know the answer, actually. Sorry.
Oh, no. Okay, it's okay. We'll just move on to the next question. Are there natural ways to target the P75 neurotrophin receptor during aging instead of
I think there
taking...
is a question before that, right? Aging affects both structural and functional processes.
Yeah,
So it
aging...
has more adverse effect on our hippocampus. So actually aging, the structural plasticity, I can say the structural plasticity is not completely affected, even though I show the difference in ROG pathway. But if you closely analyze, we observe long-term plasticity, but in a reduced situation. And we observed early form of plasticity, that is short-term memory. But the major difference was the association between short-term memory and long-term memory that was drastically impaired. So I can say the more adverse effect is not on structural plasticity, it is on functional plasticity, because the functional interaction, which is very critical for forming long-term memory, is impaired in aging. So I can say the memory, they can make memory with aging, and also in neurodegeneration like AD, in AD condition, they can make memory, but we don't know. The encoding is affected, whether the consolidation is affected or retrieval loss. Which part of memory component is completely affected, we don't know.
Yeah, I think the next question, Andrew actually asked you already. We will go on to the next one. Does our circulating levels of BDNF increase or decrease with advancing age? And in relation to these levels, what effects does it have on our memory and volume of our hippocampus?
Does our circulating levels of BDNF increase or decrease with advancing age in relation to the level of circulating BDNF which was affected? So I don't know about circulating level of BDNF, but in the brain there is a change there is a decrease in BDNF level. And for memory, we call mature BDNF as a good BDNF. In a layman style, I can say the good BDNF level is decreased or no change, but the bad BDNF level, like pro-BDNF level, is increased. So there is an imbalance in pro-BDNF to be good to bad. So that causes plasticity changes. And what effect does it have on our memory and volume of our hippocampus? So the volume of hippocampus, in some way, there are contrasting results that sometimes there is no neuronal loss. That is why in the beginning I showed some of the understanding. There are no neuronal loss and no volume loss, but in some cases they show volume change. But even though in the metaplastic aspect, it is not the volume of the hippocampus, but it's the functional connection and the functional interaction between the existing synaptic population is important for coding or making, keeping and losing of memory.
Moving on to the next question, is there a sex or gender difference with regards to the age-related changes in the hippocampal-dependent synaptic plasticity and memory mediated by the P75 neurotrophin receptor?
Yeah, this is a very good question. Actually, we started a project on this aspect. So Saki already showed the gender difference in male and female. We observed higher neuroinflammation in female AD models compared to male models. That motivated us to check what happens with P75 level. This is in progress, actually. I can't say anything at the moment.
The next question, in your opinion, should we be targeting the P75 neurotrophin receptor to combat cognitive and memory deficits during aging in humans? If so, how should we realistically do it?
Yeah, I think this is a possibility. There is a drug in our sleep deprivation paper. We used a peptide called TAT peptide, which is not completely novel. I mean, it is in the field for last 30 years, developed by Carlos group earlier. And we found that if you use TAT peptide, actually you can prevent P75 receptor mediated impairments in plasticity. So that is a possibility, but we have not yet started discussion in this aspect with the clinical studies.
Okay, for the
Are
next...
there natural ways to target P75 neurotropin receptor during aging instead of taking supplements? Natural way, I don't know, but there is, as I mentioned earlier, there is a peptide that can target the P75 neurotropin receptor, but it is not very specific. I have to admit that. But when we use this peptide, there is no side effect. I mean, the basal synaptic activity was not affected. So in a casual discussion with the clinician, he was saying that it's a nice idea to test it in human beings, but no such work is going on.
This is absolutely the case. Danica, I think we have to move to the last question. It would be great to have a supplement, of course. Danica, last question for the audience.
Okay, sure. The last question, what would be some of the major challenges / limitations of targeting the P75 neurotrophin receptor to improve cognition and memory during aging in humans?
Yeah, that's really a good question. So the animal model that we used is actually a complete knockout mice. So it's a global effect what we observe. So we are in discussion with a group from Australia to generate this conditional knockout mice. So you can target specifically on specific brain region, for example, hippocampus or cortex. So the major challenge that we face with this mice actually. So we are trying to generate the conditional knockout mice, which will give clear answer about, okay, which specific area is involved in the plasticity impairment mediated by P75 receptor. We can target amygdala. For example, Andrea was mentioning about depression. So one possibility is to check the neural network, not only in the hippocampus, and also extra hippocampal areas like amygdala, prefrontal cortex network.
Saji, thank you so much for being our guest. It was really fascinating.
I understand there is a lot of questions here which is not unanswered.
Yes.
Answered.
Yeah.
Probably
yeah.
I can, if there is a way to forward to me, I can reply to them.
Okay, that would be great. And we will put that on our website. Absolutely. Thank you so much. There are indeed lots of questions left, but we are running out of time. But please, if you have additional questions, put them in the chat. But also any suggestions are, of course, always welcome. If you would like to participate in our studies, for example, our ABLE study and a bio study are still recruiting. We're testing a couple of supplements. Please be in contact with us. That leaves me to announce who will be the next speaker on the 2nd of February, 2023, this year. Dr. Joris Dehlen from the Max Planck Institute in Germany will be our guest, and Brian Kennedy will present the show. As always, we have a final video, and listen, because you will hear lots of laughter. Wishing all our Chinese viewers a happy Lunar New Year. Take care.
为什么响头掌声在这个地方仰天大笑呢？
就是空气也很清新，然后叫了把体内的废气啊全部给排出来了。like sunshine, like springtime,
Like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.